Control L.
Hello, I'm Senay. I will present on our work, which is about an ontology-based mining of pathogen-disease associations from literature. Infectious diseases take away millions of lives every year, and this figure is from the World Health Organization's report, showing that the infectious disease, which belong to the group showing in red, and this covers communicable, maternal, neonatal, and nutritional conditions. They are the second most cause of death in the world. And we use drugs to treat infectious disease. However, we mainly fail in the treatments due to the resistant pathogens. Here, it is important to accurately identify pathogens. This will help us to prescribe the right drug and then alleviate the drug-resistant problem. And it is very important to have a resource on disease pathogen associations. For example, in order to offer more precise therapies. And recently we are working on a project that we try to develop a system or a tool that would support infectious disease diagnostic. And we aim to offer more precise therapies based on the phenotypes of patients and known pathogens. And as you can see in the figure, we link diseases and phenotypes, and then we link them to the drugs, and then we link phenotypes to disease. And then for a given patient with a set of phenotypes, we can use some semantic similarity-based method to map the patient phenotypes to the knowledge base. So there are just a couple of disease pathogen associations stored in resources. And we urgently need a reference resource on pathogen disease associations. And these associations are generally buried in literature and also Wikipedia and some other resources like Medscape. And obviously they are in unstructured format, and we needed to apply text mining methods in order to gather this information. This figure shows the system overview. Here we take around 1.8 million full-text articles from Europe PubMed Central, and then we use a well-known entity annotation tool, what is it, to identify pathogen and disease concept in text. And later, we apply a disease name abbreviation filter to reduce the ambiguity, to filter out the ambiguous disease terms. And once we have the annotated text, we employ our module to extract pathogen disease associations and store all the data in a file for the moment. So in order to identify pathogens, we use NCBI taxonomy. Our approach is dictionary-based, and it contains around 1.2 million pathogens. And we only focus on the infectious disease branch of the disease ontology. And this is around 400 infectious diseases. Our entity recognition is different than traditional entity recognitions. Here we call it as concept recognition. In traditional entity recognition, you would identify the explicit mentions of terms in text. But here, we are using concept recognition on top of the entity recognition, and once we find an explicit mention of text, we propagate the entity, or we propagate the annotation by using the ontology structure. For example, here, if you find the explicit mention of swine influenza in text, by using the ontology relation, swine influenza is also an influenza, given that it is influenza subclass, so we propagate the annotation to influenza. And influenza is a viral infectious disease, so we propagate annotation to that class all the way up to the disease. Our pathogen-disease association extraction approach is based on the sentence level on identification of the co-occurrences of pathogen diseases. And on top of that, we applied filtering, and we only focused the co-occurrences appearing more than 10 times, and then we used the MPMI association measure, and then we retrieved the ones having the value 0.2. And our set included around 56K distinct pathogen pairs, and after applying the filter, we obtained around 4.6 distinct pathogen disease pairs. And only 2.2K of these pairs were the explicit mentions in the literature, and then we obtained another 2.4K by expanding these associations. So when we analyzed the extracts, we realized that our set covers the pathogen disease associations, which would not have been possible to cover with the traditional name entity recognition system. For example, again, we found there were 2 explicit mentions of influenza, swine influenza in text, and 25 explicit mentions of influenza in text. So by using the propagation, we could associate associate 2 more pathogens, swine influenza virus and porcine circovirus 2 with influenza. There is a reference resource on pathogen disease associations, and this is KEC. So it contains around 740 pathogen disease associations, and we documented the overlaps with our extracts. As you can see, there's a big discrepancy, and the overlap is very low, is around 28%. This is not a surprising result, because we know that always literature can provide complementary results to the reference resources. And some of the reasons that we have this discrepancy are the KEC creators can use non-open access of text to create the database. But here in text mining, we are restricted with licensing, and we can use only open access part of the PubMed. We did some evaluations on our extracts. So we manually analyzed 50 pathogen disease associations, and we estimated precision as 64, recall as 84, and F-score as 73%. We look into the reasons of false positives and false negatives, and we figure out that generally we have false positives due to the ambiguous pathogen abbreviations and pathogen names. For example, in one of the articles, MPV, which is a gene name for major volt protein, it is also used as an abbreviation for metapneumovirus. So this was a false positive annotation by our pipeline. And another example, we found bronchus, which is a domain-specific name but it was annotated as an INSEC by our pipeline. So we have also false negatives due to the rejections by our pipeline, because we set some thresholds. And we also have some missing pathogens and disease names in our dictionaries. So for example, tetranychus and asthma occur only once in this given article, but it was rejected by our pipeline due to our threshold settings. So we know that the parameters which we used in our system, which are NPMI value and the number of co-occurrences, play a role in performance. However, there is no any gold standard on pathogen disease associations, so we cannot really play around with the parameters to tune the system. It's difficult. Also, we did an estimation on the overall of the performance. We didn't evaluate the subparts of our pipeline. Like we have a species annotation, pathogen name annotation tagger. So in future, we can use the Kraft corpus, which covers NCBI taxonomy annotations, and we can evaluate our system. So in future, we can also do some performance improvements by focusing on improving the precision. We can introduce a pathogen abbreviation filter to remove the ambiguous pathogen names. For recall, we can consider expanding our disease dictionary and pathogen dictionary by using other available resources. So as a conclusion, here we present an ontology-based text mining system to extract pathogen disease associations from literature, and these extracts can be useful to develop systems to support infectious disease diagnosis. If you look at the performance, it's at the state-of-the-art level when you compare to the other relationship extraction methods, and still there is some room for improvement. And in future, we would like to develop a pathogen abbreviation filter to improve the performance, and we would like to develop a comprehensive database on pathogen disease associations that would support infectious disease diagnosis. You can, actually we already started to develop this database, and you can have a look at the database from the link provided. And all the data is available in GitHub. I'd like to thank Robert, our team leader, and all the Bio-Ontology research team members for their support. And also, I'd like to use this opportunity and announce that we have available positions, and you can visit our website to get more information. Thank you.
Okay, we have time for questions.
Great talk. You did bring about the mention of the problem where we have ambiguity when it comes to abbreviations. Yeah. What do you think would your next step would be to address that one? And because this problem might grow when you have more ontologies into using this method, so there will be more ambiguities. And what approach would you use to deal with that?
To solve the ambiguity?
The abbreviation ambiguity.
The abbreviation, yeah. Actually, previously, I developed an abbreviation filter for disease names. So here, I have an additional slide on that. Maybe I can show you quickly. Yes. So here, we see that disease names can be ambiguous. Like here, you can see an example, HPS, hantavirus pulmonary syndrome, and Hermansky-Pultrag syndrome. And here, I use regular expressions to identify the long form and the short form in text first. And on top of that, I apply some rules. So these rules, like, it's checking if the Document includes also the long form. So first the abbreviation is identified, then it's checking if the long form exists, then it accepts the abbreviation. Otherwise, it filters it out. This is one rule. And second rule, if the long form doesn't exist in the text, it's checking in the ontology, it's checking if the identified long form has some specific keywords like disease, syndrome, infection, something like that. So
Thank
we
you.
can apply the similar rules for the pathogen names. So for example, for long form, we can check whether the long form includes virus or bacteria, something like that.
Thank you.
Okay.
Any other questions? So do you think that we had an earlier presentation on named entity recognition for geographic names using some word embedding and context? Do you think something like this would also help to disambiguate mentions of pathogens or diseases?
You mean to resolve the to resolve ambiguity?
Yes, and also to have some of the
Possibly.
issues where pathogens are mistaken for genes, for example.
For protein names,
Mhm.
yeah, possibly. Okay, thank you.
Okay, thank you. Okay, so our next